Yeah, thank you very much, Chris, for the nice introduction. I think also that was a very long one, and I think you got the point. I do what I do for quite a while. So first of all, thank you also for the invitation to speak here today as a keynote speaker at the American Drug Delivery and Formulation Summit. And the topic of my talk is biopredictive tools for the development of injectable drug products. So the first question that we have to ask ourselves is, when we are looking into those dosage forms, complex non-oral dosage forms, how we sometimes call them, why do we need biopredictive tools in order to develop them? And why aren't we there yet? Because many of those formulations are already there for quite a while. So I think what many people don't know is that the experience that we have with those injectables are relatively limited if we, for example, compare it with tablet formulations or capsules. That has mainly two reasons, because although they are around for a while, the first microparticle formulation composed of PLGA was approved in 1986, which means we have them for a while, but also we do not have that much of clinical experience with it that would be necessary to make it easy to develop, for example, an injectable depot formulation today. And the same is also for other formulations, for example, for the liposomes that I show here, Doxil, which was the first liposomal injectable approved by the US FDA in 1995. And this all together makes it very, very difficult to come up with new formulations and to develop the so-called IVIVC in vitro, in vivo correlations. Another reason why it is relatively challenging to develop those formulations is also that our data is limited. So we cannot just, let's say, cut out a piece out of someone's arm to find out if the dosage form is half disintegrated. And just recently, also a lot of imaging methods evolved, and that also makes it much easier to use new data sources in order to understand those dosage forms better. So the first part of my talk, I will talk about the non-biological complex drugs, which would be, for example, Doxil that you have seen in the picture before, nanomedicines and nanosimilars, and how can you develop biopredictive assays which help you also to predict the performance before you go to the clinical trial based on, let's say, the in vitro data that you can generate in your laboratory. And whenever I talk about this topic, I also want to make very clear, although I'm working in the academic field, I'm very much aware also of the different formulations that are relevant for the market. This here is a statistical evaluation that was published in, I think, 2007 by the US FDA, and we later put it into a nice pie chart where we also showed what are the different formulations that you can see today in the nanomedicine market when we are talking about those formulations. And about 1/3 of the formulations that we find in this area, the dosage forms, are liposomes, followed by nanocrystals, emulsions, and others. So you can say most of the formulations we are talking about today, when we are talking about nanomedicines and those injectable nanomedicines, are indeed liposomes. And just recently, a few new ones also appeared on the market, which are the lipid complexes that you find, for example, on Patro, which is an mRNA therapeutic, or just recently, the COVID vaccines. So when we are talking about those formulations, I want to start it with the general theory of the thing. So the question is, what makes those dosage forms release? How do they behave when we give them into an in vivo environment? Because it, of course, matters for the kind of assays that we can later use also in order to predict their performance in the in vivo setting. And one thing that very commonly happens when you inject, for example, a liposomal formulation into the bloodstream is that these liposomes initially also release a certain amount of the drug. And that has very simple reasons. You have initially a very strong dilution. You inject something right into the blood plasma. You have a huge volume. And the encapsulation, even into liposomes, is almost never 100%, which means you have a fair amount of dispersed drug initially in your formulation. And this just distributes, and you see it initially also in the pharmacokinetics, in the peak concentration in the blood plasma. But this is not the only mechanism of release. The mechanism of release describes what makes a drug come out of this carrier. And another mechanism that we very often know is the dissolution of a compound, because most formulations that we encapsulate into nanoparticles are encapsulated because they are poorly soluble. So the dissolution of the compound is also something that initially happens and that then also is capable of sustaining the release over a certain time period. And finally, when we develop those formulations, we also select the excipients in a particular way that they interact also with my compound. When you think, for example, about the COVID vaccines, we have a charged molecule and we find then a charged lipid, they basically bind together. And this is also what can affect the release, because then we have a slow dissociation of the drug from the carrier. So when we are looking at those formulations, why does it matter? Why does it matter how much release we will see over time? Why don't we look into other areas? Why don't we look into cell interactions? Why do we look into in vitro dissolution in vitro release testing? And that has a simple reason, because what we actually measure when we are looking from the pharmacokinetic side, from the clinical side or preclinical side, at the data that we get from the animal, and usually what we are targeting are the phase 1 clinical trials when we are using biopredictive assays. We want to bring it into humans, and that means this is our aim. We want to develop formulations for the phase 1 clinical trials. Then we are looking mostly at plasma samples. And plasma samples usually contain only free drug and the nanocarrier, but all the cells is basically have this interaction is happening somewhere in this invisible space that we cannot see in the plasma profile. We can only see what basically is released after, for example, nanoparticles have been taken up. And this means also that we have to look more clearly into the behavior of those formulations and how much drug they release over time. This has a huge impact on the in vivo pharmacokinetic profile later. So I want to break it down into different case studies. I promised that kind of for today's talk, and I'm very happy to talk first about phospholipid, which was the first liposomal formulation that we looked into in this context. And to basically tell you a bit of the background of this formulation, why was it developed? The first formulation of this photosensitizer, which is used in cancer therapy, it is a very, very poorly soluble molecule. It's temoporphin, so really brick dust that we are talking here today. And the first formulation that the researchers came up with, they just dissolved this compound in an organic solution. And I think everyone who is a formulator and has a bit of experience on that can probably imagine what happens when you take a poorly soluble compound, you dissolve it in an organic solvent, and afterwards you inject it into a vein. Of course, you are putting it into a strong dilution and into an aqueous environment, and the drug initially precipitates. And what you are looking at here is the pharmacokinetic profile of this first formulation, Phoscan. And initially, we do not see the highest concentration just because the drug is injected, but right away precipitates, and afterwards it dissolves over time. And that was also the reason for the next formulators that tried to improve this formulation to develop liposomes. And later then, when we wanted to establish the in vitro in vivo correlation for phospholipid, we looked into the different mechanisms of release. And what are the things that you have to consider when you are looking into liposomes and you know almost nothing about this dosage form? The first thing that we looked into, we wanted to know, does this dosage form really stay in good shape when we inject it into the blood circulation? Are those liposomes stable enough? For example, do they start to aggregate? Do we have to expect that what we are actually seeing in the pharmacokinetics is not the liposome, but something that forms later in the physiological environment when there are proteins and lipids and everything coming to those liposomes? And we measured also the stability, the physical integrity of this colloidal system. And what you see here is a measurement by NTA, nanoparticle tracking analysis. And what you can see here is that those liposomes, which have a size of about 100 nanometers, also stay in this particular size. And you see the reference in PBS, so which means phosphate buffered selene, is nothing different from the one when we actually measured it in serum, where you can also see that this particle system is very, very stable. We also do this measurement because we also want to know if over time the particle size increases, because something that is described for liposomes for a very long time, when they increase their particle size, they also have a shorter circulation time in the blood plasma. We cannot make a full prediction of the pharmacokinetics just based on the particle size. That is something that is still theory, still something to discover. However, what we can say is if the particle size rapidly increases, it will probably also affect the circulation time. And we could also say, for example, this generic product is likely to circulate shorter than the innovator product. This is something that we already can do based on that. So this first study served the purpose to see, is this formulation stable in this environment? And the second property that is equally important is the drug release. And that is also because the drug release has a huge impact on the elimination of my compound. How quickly it goes out of the carrier also determines how quickly the compound is eliminated from the human body. And a system that we use here very often in our lab, because we initially discovered it, is the dispersion releaser technology that you can also today commercially get at Matthais. They are now produce this cell, and here you can see also the commercial version. What you see here is one of my students who is injecting a pinkish solution here into the dispersion releaser, and you can see that it is built into a very conventional USP dissolution apparatus too. So you have basically a cell which is specifically made to measure liposomes in a classical compendial equipment, and this with a very, very high sensitivity. So the drug release that we measure here is basically measured by dialysis. We inject this solution into a donor cell in the center of the dissolution tester, but we do not rely only on the dialysis process. We also have a paddle stirrer, which basically agitates this solution over time and also kind of accelerates the membrane transport, because something that is very, very important when we are measuring dissolution or release of such a dosage form, we want to have sensitivity, and sensitivity means that we do not only measure the membrane permeation. What we want to measure is the release rate. And this cell was actually invented by one of my students. Here you see her, and she was really doing a very good job. If you think about that, I would be really proud if what I discovered during my PhD would later end up in a commercial product. And this is actually what she did. She developed that to the prototype, which was later then translated into a commercial product. So, and these are the kind of measurements that you can make with a sensitive release assay. And I just want to show you one measurement that we, on top of a just conventional release, do as well, because this method has a high sensitivity for separating different particle sizes. So you measure exactly the particle size that you want also to measure. You measure the particle population for its release that you want to measure, but at the same time, it also has a high sensitivity for release rates. And here we carried out an experiment where we basically compared the release from this system, basically using two different membrane pore sizes. And the background of that is that we wanted to make a separation process which basically separates not only liposomes from free drug, but liposomes from free drug from a protein-bound fraction of the drug, because we know that the amount of drug that is later bound by serum proteins also will have a certain impact on the biodistribution. And in this experiment, we used a 300 kilodalton membrane. You can see that on the top. And this membrane has a pore size which allows the drug, but also proteins to basically just permeate through this membrane. And when you compare that with another measurement, which you carry out with a 50 kilodalton membrane, here you basically have the membranes, the proteins, the serum proteins confined in the dialysis bag, and you measure from the outer compartment. Then basically what you can do, you can compare those two profiles, and from that you get an estimate of how much of the drug is released as free drug and how much is released protein bound. And that is a very powerful tool to later do also our modeling exercises. So when we are trying to understand the pharmacokinetics, and this is what we actually do, we use biopredictive tools, which means release assays, but later we have to somehow embed them also into the larger context. And this means in our case that we kind of have to also embed them into a more complex model, which gives us an idea of the pharmacokinetics in the human. And I would just want to guide you through this model quickly, which describes what happens with nanomedicines when you put them into blood circulation. Initially, we have the infusion from the dialysis, not from the dialysis, from the infusion bag basically into blood circulation, certain vascular transit until the formulation is distributed all over the vascular system in the body. And then we have the so-called carrier-bound fraction, which means the liposome circulating in your bloodstream. But because the liposomes are usually larger than the drug molecules, they only distribute inside the blood vessels, and this is also where they remain. However, at the same time, they will also be eliminated, and this is what happens by cell interaction. So everything that is lost, basically, that is not in the blood plasma anymore, we will find in this so-called accumulated fraction, which means you will find it in the RES, in the liver. You also will find cell partitioning. You will find it adhere, for example, to blood cells. But this elimination process that happens from your carrier competes with the drug release, because at the same time, your formulation will also release drug substance, and that also reduces the concentration of your drug, because what you can see here is that when the drug basically is released from the carrier, it is not in the blood vessel anymore. It will also distribute in many, many fine compartments, and this also means that it goes into deeper tissues, and this distribution will automatically lead also to a reduction of the blood concentration in the blood plasma. The yellow boxes indicate what you measure in the blood plasma, and it's actually a combination of the carrier-bound fraction and the released fraction. And when you now use this very complex pharmacokinetic model in order to make an IV-IVC from that, then this is basically what you find. So here you can see the IV-IVC that we established for phospholipid, which is the liposomal formulation, and its pharmacokinetics here. You can see that the model basically fits the pharmacokinetic curve very well. This you can see on the right, and the in vitro in vivo correlation on the left. So if someone told you IV-IVC is not possible for liposomes that are injectables because it's too complex and the RAS plays a huge role, we know that it works for phospholipid. And actually, it also works for two more nanomedicines, and one of them is the liposomal generic version of Doxil, which is Lipodox. So it actually works, and this is a work from 2021. So in this first example, what I showed to you is how we use our biopredictive assays in order to establish an IV-IVC. And this assay later can then be used to also test another formulation candidate and make a prediction of the pharmacokinetics. For example, when you develop a generic and you have another liposome formulation, you want to know how the in vivo release is. This is the way to go. So the second half of my talk will basically deal with subcutaneous depot formulations, because when we are talking about those formulations that we have here, I said initially we have two kinds of formulations that we are dealing with. One are the ones that, for example, those injectables, intravenous injectables like Doxil, and the other ones are the subcutaneous injectables, those complex dosage forms that are subcutaneously injected and that deliver also the compound over a longer time period. So, and I think what is important when you look at this particular delivery route, I think it is also important to understand what it means when you do not have this high market share, like you have it, for example, for tablets or capsules, when you have only a small market size, and what it also means from the academic side, if you basically have not let's say 100 years of experience, but you have significantly less. And this here is a statistical evaluation that we made also specifically for the subcutaneous route of administration, where we looked into the different, basically investigations that were published in the literature. And we looked also at all the different investigations and selected randomly articles and looked what is statistically, what has been used in order to predict for those dosage forms the pharmacokinetic behavior. And what you can see is that 20% of the data was generated in vitro, 50% was generated in rodents, and only 6% of the data came from humans. And this is also the literature base that you are looking at when you are reading today in a paper the assumptions that an author makes, and that is an important information when you are developing those dosage forms. But when you basically take the bottom line of all of that, what we found in the literature, the absorption from the subcutaneous tissue, it depends, for example, on aqueous solubility, particle size, molecular stability, and also the mobility of your dosage form in the subcutaneous tissue, but knowledge gaps included. That is important to understand for sub-Q. And when we are just now looking at a few interconnected model parameters that we looked into in our case studies, here I want to give you an idea how it looks like when we basically look at those dosage forms from the perspective of someone who develops a biopredictive assay. We first of all inject the formulation in the subcutaneous tissue. This is also what you can see here. You can see that we have the formulation, then we have a release process which happens, which means we have the formulation which releases the free drug, and then we have a certain diffusion rate which leads then, which basically transports the drug to the blood circulation. And one of the issues is that we only measure the drug when it reaches blood circulation. So that is an important information here. So the processes that we have is release, diffusion, degradation, and also what I didn't talk about, the lymphatic transport, because small particles can also undergo this lymphatic transport. And the first study that we did was basically, we investigated a formulation using a so-called hydrogel assay, where we basically tried to mimic the subcutaneous tissue. And what this barrier basically shows here, you can see it in the picture, it's an old study from 2016. We basically compared just the diffusion of compounds when in a hydrogel versus a study in mice. So we also used rodents here. And what we found is that diffusion and dissolution are interconnected parameters, which means when the dissolution is basically a little quicker, then the diffusion plays or makes a larger proportion of the overall absorption process. When the dissolution is slow, it does not affect the overall pharmacokinetic profile that much. Basically, in pharmacokinetics, we're talking about the rate limiting step. And this is also what my case studies will focus on. And when we are looking at the first case study, medroxyprogesterone acetate is an old compound. It is really brick dust, very, very poorly soluble. And what we found is that basically the dissolution is very, very slow, which automatically means that the dissolution drives the whole game. The dissolution is slow, and that means whatever the diffusion of the compound to blood circulation you will have, dissolution will be rate limiting. However, we found also an effect of serum proteins on the stability of the formulation that plays a role. Basically, the formulation also tended to agglomerate in the subcutaneous tissue, and that increased the particle size. However, what happened there was that basically from all the processes that I've seen before, that basically the only process that finally mattered was the release process. It was not the rest, it was not the diffusion, not the degradation, not the lymphatic transport. And if you basically want to make a prediction for MPA, what we finally found is, sorry, I have to go back. What we finally found is that for this particular formulation, mostly the dissolution rate was really what was rate limiting. And I want to compare that with my second case study. And my second case study basically shows tacrolimus, which is another compound that we also tested here. And tacrolimus is a compound which is released here from a PLGA microparticle formulation. And PLGA microparticles is a very porous material, and tacrolimus also diffuses very much out of this formulation, and it also shows significant degradation. And here you can see two different things. You can see a biorelevant release assay carried out with two different formulations, TET10 and TET21, one with a slower release and the other one with a quicker release. You can see that basically the detected tacrolimus concentration over time really shows that we have also initially an increase and then a decrease. That is because the compound really is released and afterwards it degrades again. And at the same time, you can see what kind of model assumptions we had to make in order to basically simulate this process in order to use later also the release for a good prediction. And here you can basically see that we have a significant impact of the lymphatic uptake, the degradation in the subcutaneous tissue, and the diffusion. They are basically overlapping processes. And particularly because we had here a significant diffusion time, we also have here an interplay between the diffusion of the compound, because the slower the compound diffuses through the tissue, the more time the compound has also to degrade. And that is also a very serious issue because it makes this background correction very, very difficult. However, we finally managed also to do that in this context here, to basically deal with tacrolimus as a compound. So if we are looking in this particular process, you can see we start again with a formulation. We have here those interconnected model parameters. We have the release, the diffusion, the degradation, and the lymphatic transport, all of them involved in the overall pharmacokinetic profile. And that is also what plays here together. However, I will basically just tell you what in the end we found out when you balance those processes, when you weigh them against each other in a pharmacokinetic model that we used here. You see here the model structure. Then basically, finally, what we found was that the in vitro in vivo correlations that you can achieve by just using the right tools on the pharmacokinetic side together with the right tools on the release side, those two together give you a very, very good estimate of the in vitro of the in vivo performance in an in vitro setting. So, and finally, after basically telling you about the two areas that my group is also basically exploring in our research, I want to finish this talk with a take-home message for you today. I think I made very, very clear that complex non-oral dosage forms exhibit different release mechanisms from other formulations. And this also explains why very often we have significant difficulties when we are developing those dosage forms, because we do not really foresee what happens in the subcutaneous space with those formulations. And that is a problem in the development. Performance generally, I do not really like the term dissolution testing. I like the term performance testing because performance is a multifaceted process. It can depend on degradation, it can depend on diffusion, it can depend on dissolution. And mimicking selected mechanisms can sometimes be quite successful. Here I want to point out this example, medroxyprogesterone acetate, a very poorly soluble compound, which is then very slowly released. Dissolution is rate limiting, so why should I measure diffusion if it doesn't matter in the in vivo context? But what does it require to develop a good biopredictive performance assay for those formulations? That's my final question here. And I think what you have to know what you are testing, because the formulation very much affects what kind of release mechanism you will see, and that automatically affects what kind of performance test you need for your dosage form. Okay, with this, I say thank you very much. And in the end, I always show also this video. I'm not sure if that now starts playing, but you can also find it on YouTube. My group recorded that, where basically the people who really did the work also have a very short, basically run through our lab. So you can find that. And if you want to know more about us, then you can find us also on Instagram, Twitter, and so on. Thank you very much. And I'm very much happy to answer your questions if there are any.